your opening remarks, if that's all right.
Yeah, that's fine. Why don't you go ahead of me?
Okay. I just wanted to make sure all the graduate students know that we've got a number of graduate student awards available in the Department of Plant and Soil Sciences. So at the end of today, I'll be sending out a Qualtrics tool that you and your advisors and even faculty member who are not your advisors can use to nominate you. We've got the Mott Award, which is a Society Award that's open. We've got the Johnston Carringer Award. We've got the Philip Peasley Award, and we have the DeCroney Seed Biology Award. So I'll send that to the grad students and to the faculty today. And so just have that on your radar. Those will be due February 26th is when we're going to make them due. So please take advantage and apply for those. Thanks, Jason.
Thanks. I don't have a huge amount of opening remarks, so I'm just going to wait about 5 minutes and then we'll get started. Virginia, are you able to share your screen?
Yes. Hello.
Okay.
Let me see.
Okay, great. Audrey, do we have any opening slides?
We don't have any opening ones that I know of.
Okay. Okay, I guess we'll go ahead and get started. Welcome everyone to the 2021 Graduate Student Integrated Plant and Soil Sciences Graduate Student Symposium. This is obviously a little bit different like everything else in our lives, but we managed to pull it together. And I'd like to start out just by thanking everyone who made it possible. First of all, I'd like to thank Emeritus Professor Bill Witt and Don Sparks, who's an alum of the department and now a professor at the University of Delaware, for their generous gifts that make this symposium possible. I'd also like to thank Dr. McCauley and the Department of Plant and Soil Sciences for providing resources to make it possible, including staff time. Abby Kane and Audrey Sparks, thank you so much for helping pull this all together, pull together the scheduling and the judging forms, abstract book, all the technical aspects. You all just make life so much easier for us, and we really appreciate you. So for those of you that don't know, starting last year, we went to a once per year symposium. I know some of you are new here. And thank goodness we were able to have a major blowout at Commonwealth Stadium last year. Little did we know at the time that the virus was already circulating. I'll be, in our homes visiting electronically the next year. So it was really nice that we were able to do that and have really great memories of that. So we went to this once a year format. And the way it works is the students from PLS 772, which is the course that we called Seminar in Plant and Soil Sciences, or the organizing committee. And so those students take the course in the fall, and we practice presentation skills, oral and poster presentation skills, and then talk a little bit about sharing organizing symposia and chairing sessions. So the students from that course are going to be the moderators of the poster session and the oral sessions, and they've helped organize sessions. So those students are Zach Bird, Jared Cochran, Amber Herman, Annie McGraw, Kent Pham, Taj Beer, Raihan, Brian Reinhardt, Hannah Somerville, and Ella Suleta. So thanks to all of you for participating in the course and helping organize this. And you'll be seeing some of them throughout the day. Just a little bit of housekeeping. The symposium is going to be organized into 4 oral presentation sessions with 5 presentations each. The presentations are 15 minutes, including questions. So the students are going to be aiming for 12 minutes of presentation time and 3 minutes of questions. And then we're going to have a morning break in between sessions 1 and 2 for 15 minutes. And then during the lunch hour from 12 to 1, we're going to have a virtual poster session. So with your invitation, you should have received the abstract book and a link to the electronic posters, which have been available for a couple of days now. So you can view the electronic posters. And then I invite you and encourage you to interact with the poster presenters by entering individual breakout rooms that each poster presenter will have during the session. So, you know, grab your lunch, sit down, check out some of our wonderful posters and interact with our fantastic students. So I want to thank all the students who have submitted their abstracts and participated even during, you know, these unusual times. And thank you so much to all of our faculty judges who've volunteered to their time to give our students valuable feedback on this. So with that, that's really all of the remarks that I had to start out. So we have a few minutes. Virginia, if you want to just kind of get situated, it looks like you've got your slides sorted out, and then I'm going to hand it over to Jared. So Jared, the session is yours.
Okay, thank you, Jason. Can everybody hear me? Okay. Hi, my name is Jared Cochran. I'll be the first moderator for this session. Just so the speakers will know, at 10 minutes, I'll come on and just say 10 minutes left, letting you know that you have about 5 minutes to finish up your talk. Virginia, are you ready to go?
Yes.
Okay. So first up, we have Virginia Vargas. She works for Dr. Van Sanford, and she will be presenting on the genomic selection for Fusarium head blight resistance in wheat. Okay, Virginia, if you're ready.
Okay, thank you, Jared. Thank you everyone for being here today. The title of my presentation, as Jared said, is Genomic Selection for Fusarium head blight resistance in wheat. Genomic selection is a plant breeding tool that uses statistical modeling to predict how a genotype will perform without field testing. And how does genomic selection work? The central process in genomic selection is to calculate the genomic estimated breeding values for genotypes for selection candidates that have only genotypic information. This is done training a model with individuals that become the training population that do have phenotypic and genotypic information. After the model is trained, we get the genomic estimated breeding values, and the breeder can make selections and advance lines in the breeding program. How do we know that our model is working? Calculating the prediction accuracy. Prediction accuracy is a correlation between the genomic estimated breeding values and the phenotypic values. What we want is to maximize the prediction accuracy for the set that is being predicted. Many factors affect prediction accuracy. Some of them, the most important, are the number of markers, the training population size, and the optimization method, for example. That is the methodology we use to select the training population. Genomic selection became a very promising tool when breeding for disease resistance because the most worldwide challenging diseases have quantitative inheritance. That means that the traits are controlled by multiple genes with major and minor effects. So, breeding for quantitative resistance require multiple cycles of breeding. Because of this, genomic selection becomes ideal for these complex traits because the model have great power to capture small effect loci. We work with Fusarium head blight. The disease Fusarium head blight is a devastating disease in wheat and barley. It's caused by Fusarium. It's a very damaging disease that reduces yield, reduces test weight, and produces a mycotoxin, deoxynivalenol. This mycotoxin contaminates the grain and could be toxic for animal consumption and also humans. The symptoms of this disease, as we can see in this photo, is the premature bleaching of the spikelets and the tombstone. The tombstone is the disease grain, as we can see in this photo in the bottom. The tombstone is the disease grains on the right, shriveled seed with the pinkish mycelium. And it's a big problem and affects also the commercialization for farmers. One of the strategies that has been done to fight against the disease has been the establishment of regional Scav nurseries. In the eastern part of the US, we have the Northern Uniform Scav Nursery and the Southern Uniform Scav Nursery. This is a public and private breeding effort where breeders send lines to many other locations every year, and that breeding lines are tested for the different Fusarium traits, and this generates information not only for the breeder, also the information is available for all the breeding community. And this has helped increasing the levels of resistance. So with all this said, the objectives of my presentation of this study were to use the regional nurseries as training populations to predict Scav resistance in lines from the UK wheat breeding program, investigate the training population design, the training population size, and the training population optimization method, and evaluate the effect of reducing the marker number used to run the model through GYS, a genome-wide association study. So in the first study I'm going to show, we have used the regional nurseries as training population, like is shown here. The training population became the regional nurseries with 4 training population sizes and 3 optimization methods. We obtained phenotypic data from these nurseries 5 years for 5 traits. We are going to focus on 3 traits today: DON, the mycotoxin accumulation, and 2 indexes, DSK and DK, and the genotypic information, the genotypic data to run the model were 20,000 SNPs. The selection candidates we are going to predict are Kentucky lines evaluated in 2017 with the same amount of markers. So the phenotypic data for these studies was collected during 3 seasons in Lexington, Kentucky at Spindletop Farm in an irrigated and inoculated nursery for all the traits that we measure every year. And the genotyping was, and also the marker imputation and filtering was performed in the USDA genotyping lab in North Carolina. So to test the predictive ability of the model, we first ran 100 cycles of cross-validation for the 3 traits I was mentioning and for the individual sets, the Northern Nurseries, Southern, the combined nurseries, and the Kentucky breeding lines. Prediction accuracies were moderate, ranging, for example, for DON from 0.49 to 0.6, and for the other 2 traits, similar values from 0.5 to 6.4 and 0.1 to 6.4, which is a good outcome that our model was working. Next, we calculate the prediction accuracies for the same 3 traits when the regional nurseries, NUS plus FUS combined were the training population at the 4 different training population sizes and with 3 different optimization methods. As an overall conclusion, we had a significant effect of the training population size, meaning that we find a good response that at increases of the training population size, the prediction accuracy increased for the traits. In general, we didn't find much differences between 300 and 400. These were the 2 training population sizes with better outcomes. DSK was the trait with better prediction accuracies. Here we see 0.8 and 0.9 for the 3 optimization methods. And we did not find a significant effect of the training population optimization method for the 3 traits. So we analyze next the implication from a breeder perspective of these results. Here we can see the proportion of correctly selected lines based on breeding values for the for the 3 traits under 2 different selection intensities, 20% and 40%. This means, for example, that for an important trait like dawn, based on the breeding value for dawn, 44% of the lines were correctly selected based on predicted values compared what would be selected based on the phenotypic values, with a selection intensity of 20%. And being more conservative, that means keeping lines for further evaluation, we found out, for example, that with predicted values for DSK, this index, almost 70% of the lines were correctly selected for low dawn compared to what would be selected with the phenotypic data. In the second study, the regional nurseries are again the training population. We are going to focus in 2 traits now. The selection candidates are 3 sets of lines evaluated in different years, Kentucky lines. And what we did here to investigate the effect of the marker numbers was to find trait marker associations with a genome-wide association study, and we could identify significant SNPs for dawn and DSK, and that with that significant SNP, we built 3 marker scenarios, 2, 3, and 4 at different p-values. And with these marker scenarios, we explain the model. Here in table 1, what I want to show is how the marker number was reduced. Really a very big reduction from the 20,000 in scenario 1 to this new set, these new marker sets. So in table 4, we can see the prediction accuracy, again with cross-validation to test the model, where we have for 2 traits, dawn and DSK, prediction accuracy under the 4 marker sets scenarios. We found that the scenario with the full set of markers, the full number of markers, we obtained the highest prediction accuracies, and the prediction accuracies with the other 3 scenarios was slightly reduced. But again, moderate prediction accuracies. And this slide shows for the 2 traits, for dawn and DSK, the prediction accuracy under the different marker sets and at 2 different training population size. So here what we see as an overall conclusion is that at TP, at the training population of 100, the scenario 4 obtained the highest prediction accuracy. For the 2 traits, that was around between 12 and 20% higher than the scenario 1, the full set of markers. And at a training population size of 400, we found the highest prediction accuracy with the full set of markers. And this was around between a 3 and 10% higher than the second best scenario, that was again a scenario 4. This was for the average of the 3 years of selection candidates. So the conclusions of my presentation are that the use of regional nurseries as training populations was successful with moderate prediction accuracies. A training population size of between 300 and 400 individuals show the highest prediction accuracy for all traits. The different optimization methods did not show significant differences for all traits. A genome-wide association study offers an excellent tool to select significant markers for traits like DON and DSK. Marker set selected at a p-value of 0.1 show the best performance. These results, finally, help the breeder to find the best way to allocate the resources within the breeding program, reducing costs and time in the breeding process. So with all said, I wanna thank you, the department, thank my advisor, Devan Sanford, Dr. Gina Brown, that was in charge of all the genotyping, the wheat breeding lab, and all the students that helped us during the summer. Thank you.
Thank you, Virginia. We have time for 2 questions. Okay, well, I have a question. Can these biomarkers be used to predict resistance against like other diseases, or is it only useful for Fusarium?
No, this can be, yes, model can be used for any disease or any other trait. The important part is that you need to have in the training population individuals with the disease or the trait that you want to predict. But you can predict any trait, height, morphological traits, other disease traits.
We have a question. What would you suggest breeders in terms of cost? Full set of markers or selected set?
Yeah, that was one of the, I would suggest with these results, I would go with a selected reduced set of markers, because I think that what we have seen here in prediction accuracy is very small, and you really can reduce a lot what you spend in markers, in genotyping.
Okay. Well, I think that's all the time we have. Thank you, Virginia.
Thank you.
Okay. Next, we have Elzbieta. Are you ready to present?
Jared, I was thinking maybe we should wait we're a little bit ahead of schedule, and I'm worried that if someone was coming in to see a certain presentation that they might miss part of it.
Okay.
Why don't we wait until 9:30?
So Jared, I noticed Virginia has a question in the chat box. Can you see the chat box? You might.
Now, yes, wait, let me see.
Yes, that's the question I read out.
Oh, sorry.
Ella took the pictures. She's the photographer of the team.
Maybe Ella could go ahead and share her slides, her screen, and then we'll be ready to go at 9:30.
Yeah, can you hear me?
Yes.
Okay, finally.
I'll give you just about 5 minutes to stew in anxiety before presenting.
I just want to make sure that nobody misses a presentation that they were logging in to see. think we have significantly more people for the morning session than we have in the past. I guess the commute from the kitchen table to the computer is a little bit easier. If you're just logging on, we're just having a short pause here to get back on schedule.
I guess I can let you all know that we did send the links to Bill Witt and Don Sparks, and unfortunately Bill communicated that he had a conflict for most of the day, but Don said he may join in when he can, so he might be popping on and off. So hopefully he gets to see a little bit here.
I didn't see him on. So anyone notices that he's logged on, say something so that we can acknowledge him.
I'm presenting. We just have a couple minutes because we started early.
Okay. So it's about 9:30, so we could probably start again. So next up we have Elzbieta Zuleta, who worked for Dr. Phillips, and she will be presenting on the evaluation of winter rye sea cow cereal for adaptation to Kentucky. Okay, if you're ready. You're still muted.
How about now?
You're good now.
Okay.
Okay.
Okay, good morning, everyone. Today, I'd like to present you some results of evaluation of winter rye for adaptation to Kentucky. Many times I heard the question, why we have such a problem with rye growing in the United States, and I didn't really know the answer. And finally, one day I found this interesting article that helped me understand what is actually going on here. First of all, rye has a rich heritage in the United States. Maybe you know that the first president of the United States was a rye distiller, and rye used to be a truly American spirit even before bourbon. Unfortunately, a set of historical events, especially the Prohibition, caused a decline in rye demand and the decline in support for rye research. As a result, over 50 years, there was very little work done in rye breeding. At present, the interest in this crop is increasing thanks to its wide range of use, pro-health properties for human nutrition, for its specific flavor and spice for whiskey that is not possible to get from any other grain. Unfortunately, rye production in US is not enough to meet the demand. So to increase rye production in Kentucky, we need to encourage farmers for growing this crop by introducing them well-yielding variety that will suit Kentucky environment and bring them benefits. We conducted planting dates experiment. Experiment was planted in UK North Farm in Lexington and at Darreville in Western Kentucky. Planting was done in 2018 and 2019. In each environment, we tested 24 varieties of winter rye in 3 planting dates, early, middle, and late. And here is the table that is representing our choice of varieties that was based on the availability on US seed market. And after first season, we noticed that some varieties did not work well in this environment. So we decided to replace them for next season with others. Planting dates were planned to be approximately 2 weeks apart. Of course, the final planting dates were determined by weather conditions and the logistics of field work. And in 2019 fall, we experienced very unfavorable weather and early planting in October was not possible to do. So we ended up having early and middle planting in Western Kentucky, only 2 days apart. Pre-harvest measurements that we took were heading date, Fusarium, septoria, leaf rust, and lodging resistance. Heading date was marked as a date when 50% of heads of single plots were visible. Disease resistance was evaluated visually at 1 to 9 scale, where 1 was standing for susceptible and 9 was standing for resistant variety. And after harvest and collecting yield data, we chose 18 varieties from 2019 harvest and 10 varieties from 2020 harvest for grain quality and predicted alcohol yield. The grain quality analysis included moisture, protein content, and content of seed fractions. And predicted alcohol yield was presented as liters of absolute alcohol per ton of grains. We've screened a wide range of varieties with different origins. We had hybrid varieties from Europe, varieties from Canada, and various states. We also tested Kentucky lines that were locally developed by Dr. Tim Phillips. And these pictures are showing that for some varieties, Kentucky environment did not work well. On the picture on the left, taken in mid-December, you can see much taller varieties, which were Florida 401 and Merced, that were growing very fast in fall and tall, and they got cut by frost. And on the picture on the right, you can see how damaged were these plots. They were completely devastated by freeze. Earliness was assessed as a number of days from January 1st to heading date. In this graph, we can see the mean number of days for each genotype over all environments. And we can see that genotypes planted early were heading earlier, and genotypes planted late were heading relatively later. There were some exceptions, like Florida 401 and Merced, the two varieties that got damaged by frost. They were heading at very similar time in all planting dates. The peaks were reached by hybrid varieties, Braceto, Danielo, Bono, and Serafino, and also population variety Aventino, which is grown by farmers in Kentucky, and higher yielding Hazlet and Dillon. If we look at the group of KYSC, these are the Kentucky lines developed by Dr. Phillips, and they were selected for Kentucky environment, we can see that they were relatively early comparing to other varieties. But if we look at mean yield of all genotypes, we can see that the number of days to heading was much lower in Western Kentucky comparing to the data from Lexington over all planting dates. This bar plot is representing genotypes mean yield in bushels per acre. And we observed high variability in yield in both planting seasons. But both seasons, we've seen that the highest yielding varieties were hybrids, Braceto, Bono, Serafino, and in 2020 only tested Danielo. And out of population varieties, the highest yielding were Hazlet and Dillon. These graphs are showing the mean yield of all plots harvested in each location over planting dates. And we can see that in general, all the yields were higher in Western Kentucky, marked as a dashed line on both graphs. Also, in 2020, we experienced severe freeze in May, and early planted in Lexington suffered the most from this condition. So we didn't even harvest that block, and this is why the data is now not shown on this graph. As I mentioned, 2020 plantations experienced severe freeze in May, and that caused a lot of damage, but also led us to very interesting observations. As we can see on this figure, almost all varieties performed better in Western Kentucky, shown with dashed line. And this may be because of the difference in latitude and different maturity of the plants at the time when Kentucky experienced May freeze. Plants in Western Kentucky already started producing kernels at that time. And in Lexington, plants were at anthers extrusion stage, so delicate anthers got damaged by freeze. They were producing very little pollen. Plots were less fertile, so as a result, yields were much lower in Lexington. Something that we are looking at when it comes to distilling industry is alcohol yield. And thanks to Center for Craft, Food and Beverage of Hartwick College, we were able to compare our varieties in terms of predicted spirit yield. We've seen that in general, higher predicted alcohol yield was assessed from grains harvested in 2019. And we also learned that even though
10 minutes.
hybrids were yielding at highest levels, the highest spirit yield was predicted for population variety in 2019. But in 2020, the winner was hybrid variety, Bono. These figures are showing the relation of protein content and predicted spirit yield, and relationship between plum seeds content and protein content on the bottom figure. And we found very strong correlation, negative, with protein content and predicted spirit yield, and moderate positive correlation between plum seeds content and protein content. As conclusions, we observed a wide range of maturity among tested varieties. We know that farmers prefer to grow earlier varieties for logistics reasons, but the highest yielding varieties that we tested were hybrids which are late. High mean yield of all genotypes was achieved in Western Kentucky, and this suggests that rye requires more days for development and grain production. Different varieties performed better at different planting dates, but in general, in 2018-2019 season, the best planting date was early. But in 2019-2020, the severe May freeze caused a lot of damage, and that season, the best planting date was the late one. We found negative correlation between the predicted spirit yield and protein content in grain. And we've learned that high grain yield does not always relate to high spirit yield. So there must be found some economic balance for alcohol production. For further steps, we started building a breeding base, selecting 140 lines from gene bank for earliness and general performance. The selected lines will be evaluated for aroma and grain compounds responsible for aroma and flavor. We are trying speed breeding method to speed the breeding process, and we are conducting economic study to establish the most beneficial approach for rye production. I'd like to acknowledge my advisors, Dr. Van Sanford and Dr. Timothy Phillips, Dr. Chad Lee and Carrie Knott, who are very helpful with this project, our wheat breeding crew, and the founders of this project. Thank you so much for attention, and I'll do my best to answer your questions.
Okay, thank you. We have time for one question. Okay, we have a question on comment.
Can you
And
read
chat.
it?
Yeah, based on differing results, what is your recommendation for planting time?
For now, I would say early. Yeah, we've seen that this past season, the best was late planting, but this was due to very unexpected weather in May. So based on the previous season, I would for now.
Okay. I think we have time for just maybe one short question.
Mm-hmm.
Why might the higher protein content have the negative impact on predicted spirit yield?
Oh, first of all, the higher protein content is, the lower starch content is, and the starch is the main thing that is needed to produce alcohol. So the more protein, the less starch, and the less alcohol yield.
Okay. I think that'll be all the time we have for questions. Thank you again. Next up, we have Amber Herman. Okay. So next speaker is Amber Herman. She works for Dr. Lagleiter. Her presentation will be on the evaluation of Italian ryegrass, Lolium prion multiflorum, seed retention and distribution in wheat at harvest. So Amber, if you're ready to go.
Okay. Hi, my name is Amber Herman, and as he said, I'd like to present the results from my experiments where I looked at evaluating Italian ryegrass, Lolium prion species multiflorum, its seed retention and its distribution in wheat at the time of harvest. So a little bit of an introduction and overview to my project. I'm going to introduce to you what Lolium multiflorum is as a species, and then give you some background information on harvest weed seed control technologies. And from there, I will talk about the objectives of my studies. Then there's going to be two parts to my experiment. The first part is a pre-harvest seed rain. I'll give you the materials and methods, my data collection, the results, and any conclusions. Then the second part of my experiment is at the time of harvest, the seed distribution. I'll go over the materials and methods, data collection, results, and conclusions again. And then tying the two together, I will give you some overall conclusions, and then I'll show you where I'm taking this in the future and take any questions. So Italian ryegrass or annual ryegrass is Lolium prion species multiflorum. It's a weed that's increasing in prevalence in its Kentucky wheat fields. Recently, there's been an increase in complaints in post-emergence herbicides in wheat, and there is some known resistance in Lolium multiflorum. So we know there is some issues with that. There's some ACC inhibitors, there's ALS inhibitor resistance, and then recently through UK, we've also found some more ACAs inhibitors, panoxidine, and then also maybe glyphosate. This is something else I'm testing on, but that's not what I'm presenting on today. With all this resistance issues, it has been found that there is very few post-treatments left for Lolium multiflorum, and as a result, farmers are having to rely solely on pre-applications. So Lolium multiflorum and wheat grow at the same time. Therefore, they're going to compete, which can lead to less wheat yields. Lolium multiflorum is going to germinate at the same time as the wheat, so they're going to grow the entire time together. And for a single Lolium multiflorum plant, for 9/100 of a square meter, your wheat yield can be reduced by 4%. So it doesn't take a lot of these in your field to reduce your wheat yield greatly. So harvest weed seed control is an emerging method to deal with weeds that have herbicide resistance issues. It's becoming widespread as more and more weeds have herbicide resistance. In Australia, they use roller mills attached to a combine to destroy any weed seed in the chaff that exits the combine. In a study out in Australia, they found with Lolium rigidum, which is a cousin to Lolium multiflorum, it has many herbicide resistance issues, and it's something they're struggling to control in wheat. And they found that with the roller mills attached to the combine, they controlled or destroyed about 95% of the seed in the chaff, which means it's not going in their seed bank, which means it's not going to germinate, and therefore it's not going to be a problem. So the objectives for my two studies is to evaluate the seed retention and rain of Lolium multiflorum prior to wheat harvest. And then at the time of wheat harvest, evaluate the seed shatter and dispersal of Lolium multiflorum. So looking at my pre-harvest experiment, my hypothesis is Lolium multiflorum will retain its seeds up to the time of wheat harvest in Kentucky wheat fields. So I have four sites, but you're only going to see data from the first two today. The second two, which have the asterisks, Warren County and Russellville-Gallatin Road, I have these samples, but I've not got through cleaning them and analyzing them. As you can see, that there's two different sizes of fields, and therefore we took a different number of samples, and these were collected roughly in the middle to late June. All these sites, though, were evaluated in mid-June up to the time of wheat harvest. We were looking for fields with very heavy infestations of Lolium multiflorum. And then when we collected our pre-harvest samples, they were all harvested roughly a few days, if not the same day, before the wheat actually was harvested. We know the UKRAC site was harvested a day later. The Warren County site was harvested the same day, and the Young Road site and the Russellville-Gallatin Road sites were harvested a few days later. So looking at my materials and methods, we looked at a meter squared for each roughly 0.2 hectare of infested field. So what you can see in that picture is we took a meter squared, and in that meter squared, we collected all the Lolium multiflorum seed heads and counted them to determine the density of the infestation in this field. Also in that meter squared, we collected wheat heads to kind of get an idea of where the grain moisture level was at. And then lastly in the square, as you can see in the picture, we used a shop vac to collect any seed, chaff, or debris that was on the soil surface. Now going over those samples, they had to be cleaned. So the seed heads, they were all hand thrashed then and then put through an air column, seed blower. As for the ground samples, we used sieves to separate any larger debris or finer soil. And then lastly, we used the air column seed blower to clean up the rest. Now, from those samples, then we took 10 randomly selected ones, weighed out a 1/2 gram of seed, counted the seed in that 1/2 gram. We did this for both the ground and seed collections. The rest of the samples were all weighed, and the counts from those 1/2 gram samples were used to estimate the seed in the rest of the collection. Here are the results then. These two sites were analyzed separately, therefore you see lowercase and uppercase letters. As the young road site, you can see the amount of seed on the seed heads was significantly different than what was found on the ground. There was roughly about 11,000 seeds found on the seed heads versus about 2,500 found on the ground. As for the UKREC site, the same thing was found. It was significantly different that there was more found on the seed heads than the ground. There was roughly about 4,000 seeds found on the ground or that had already shattered. And again, there was about 11,000 seeds still found on the seed heads. Now, just to give you an idea of what these sites were like, at the Young Road site, we found an average of 77 seed heads per meter squared. And at the UKREC site, we found an average of 125 seed heads per meter squared. And the average grain moisture then at the Young Road site was 23%, and the grain moisture at the UKREC site was 16%. Now from this, we can draw on the conclusion that the majority of the Lola multiflorum seeds are going to be retained on the seed head up to the time of wheat harvest in Kentucky. And this was for the summer of 2020. Now going on to the second part of my study, I'm looking at the time of harvest, and my hypothesis for that is the majority of Lola multiflorum seed will pass through the combine then and be found in the crop chaff. So for this study, we used the Research Farms Combine. It's a 9650 John Deere with a 7.62 meter head. The fan speeders run at 1280 RPM. The cylinder speed was 830 RPM. The concave setting was 1, and the speed was 5.6 kilometers per hour. This is not what we set it as. This is what the farm crew would run it as. And we wanted to do this because this is what the average person would run it at for this field. We wanted to see without any implications, because maybe going forward, we might be able to change it, but this is what it was ran at that day. So then looking at this, we wanted to see where the seed is distributed at the time of wheat harvest. We were looking at whether it shatters at the header, if it goes into the chaff collection, or it's going into the grain tank. In this study, there was 4/10 of a hectare blocks. From those blocks, 4 subsamples were taken for the head shatter and chaff collection from each of the 4/10 of a hectare plot. And then to get the head shatter and chaff collections, we used trays that equaled a meter squared, so everything was the same. For the head shatter, we had 24 trays. They were roughly 25 centimeters long by 17 centimeters wide, and they were laid within the wheat rows. The combine was allowed to come to full speed as it was harvesting. It came up to our trays, and at that point, we had the combine stop and back up. Whatever was in that trays, we collected as our sample. And that was done 4 times. For the chaff collection, it was similar. There was 4 larger trays, 66 centimeters by 46 centimeters, that were placed behind the combine. For this, there was two people. There was one on each side of the combine, and it was allowed to come to full speed to get harvesting again. Once the header had passed them, they laid down their 2 trays on each side of the combine, and the combine continued to harvest past them. Once there was no more chaff being collected in these trays, the combine again stopped, and from those 4 trays, we collected everything as our sample. Again, that was done 4 times per block. Lastly, at the end of each block, the grain sample was taken using a chambered grain probe. At the end of each block, after we took our sample, the grain tank was dumped, so therefore there was no contamination from block. Now, the cleaning and counts were done similarly to the pre-harvest study. For the head shatter, we sieved out the samples to eliminate any larger debris or chaff and finer soil, and then finally they were cleaned using an air column seed blower. The same was for the chaff collection. The samples were again sieved, but to eliminate any larger chaff and finer materials, and then again put through an air column seed blower. Lastly, for the grain tank, we just use an air column to separate any wheat and the Lola multiflorum seeds. The counts again were done the same. We picked 10 random samples, weighed out a 1/2 gram of seed from those subsamples, counted this seed in that 1/2 gram, and we used those counts then to estimate the rest of the seed in the collection. And then the rest of the samples were then weighed. So these are the results from the harvest seed distribution. And as you can see, it was interesting. We found that the chaff collection was significantly different from what was found at the head shatter. There was roughly 7,000 seeds found in the chaff collection and roughly about 4,500 seeds found in the head shatter. Now, the grain tank was not significantly different though from what was found in the chaff collection or the header. There was roughly 5,000 seeds found here. And again, just a little reference for this site, the UKREC site had an average of 125 seed heads per meter squared, and the grain moisture there was 16%.
10 minutes.
Thank you. So conclusions for the harvest seed study then. It was found that a greater number of Lola multiflorum seeds were found in the combine chaff than what are lost at the combine header. The seed in the grain tank though was equivalent to both what was found in the chaff and what was shattering at the combine head. Therefore, we're thinking combine adjustments could be made to redistribute some of that Lola multiflorum seed from what was found in the grain tank to coming out the back of the combine in the chaff. Pulling from both studies now to get some conclusions, we found that the majority of the Lola multiflorum seeds were retained on their seed heads up to the time of wheat harvest in Kentucky in 2020. The majority of the Lola multiflorum seeds were either found in the combine chaff or the grain tank, which means they are entering the combine. The shatter of the Lola multiflorum seeds at harvest was found, but it was proportionally less than what was found in the chaff. And lastly, this is kind of leading up to that harvest weed seed control is a potentially viable option then for Lola multiflorum in Kentucky wheat because most of that seed is entering the combine. Going forward, there's 2 additional sites that I have to clean and analyze yet that will be added to this data. We're going to repeat the seed retention and harvest study this summer in 2021. And then this leads to the possible investigation of maybe using some harvest weed seed control techniques at the time of wheat harvest. So I want to thank everybody in the Legletter Weed Science Lab for helping with this. And I want to thank the Kentucky Small Grains Growers Association for funding this project. And I will take any questions.
Thank you, Amber. We have time for questions. We have a question in the chat. Are combined rollers available here in the US?
Yes, we were looking at getting a seed terminator from Missouri. It didn't happen this year, just things didn't work out. So that's what we're trying. We're going to try and use. Hope it's possible this summer that we could have a combine with the roller mills attached that we could see that since the seed is entering the combine, would this actually work as it does in Australia?
Okay. Any other questions? We have another one in the chat. Are there ways to handle the chaff, such as composting, that would render the seeds unviable?
We're not looking at that and we don't know, but there is something that we're looking at with chaff collection to see if water hemp can be decomposed in chaff lines. But as for ryegrass, I don't know of anything that could be done for this in wheat.
Okay. We still have time for questions. One question I had was, are the rollers only good for lolum, or have they been looked at for other plant pests?
I know in Australia they're mainly used for Lolum rigidum. Therefore, that's why we're trying it with Lola multiflorum because they're similar seeds. But over in some other states, they're looking at using it for water hemp and soybeans. So it is being evaluated for other weed seeds in the United States.
Okay. We have time for 1 or 2 more questions. Does the roller work by just squishing the seeds?
It's basically a combination of grinding the seeds and there's blades that kind of chop up the seed. Therefore, it's rendering it destroyed, whether it's kind of sliced up or grinded.
Okay. Do the blades have to be replaced after some time, or?
That's a good question. And I don't know. I assume so after so long, if they're become dull, they're no longer doing their job, that they would have to be replaced and or sharpened.
Okay. Okay, well, thank you very much, Ember. I think you can prepare for the next speaker. Dan, if you're ready.
Yeah, let me. All right, you all see that? Hear me?
We see that.
Hear me good?
Yes, I can hear you.
All right.
Okay, everybody. The next speaker is Dan Quinn. He works for Dr. Lee, and he will be presenting on corn response in furrow fertilizer and fungicide across rye covering crop termination timings. Okay, Dan, if you're ready.
All right. Thanks, Jared. So farmers in Kentucky really are driven to protecting their soils. I think that comes from why no-till began here. And it really stems from the soils being so shallow here and the rolling hill topography that we have. This picture here kind of really highlights that. These are 3 soil cores we took this last year with a hydraulic soil probe, and we can only get about 20 centimeters. That's pretty prevalent across the state. So farmers really want to take any means necessary to protect their soils and build their soils. And that's where rye cover crops come in, especially cereal rye. Rye is the most common species for cover crop in grain fields in Kentucky, really because it's on the cheaper side. It does a really good job of growing anywhere and producing a lot of biomass. So it really helps with the soil erosion. It takes up a lot of nitrogen to help with nitrate leaching. It can help with resistant weed populations, water retention, and again, building those soils up with soil organic matter. Well, the question, a lot of farmers have and producers is, how do we maximize the benefits that a rye cover crop can bring? How can we push the rye to really help with erodibility and soil organic matter? And really, one of the best ways that have been found to do this is altering the termination time, or when you spring terminate that rye cover crop. And that's kind of what we'll focus on throughout this presentation, what we did in this study here. So this is from at Lexington this last year, and you can kind of see the different termination timings. Rye on the left was terminated about 1 day after planting, and rye in the middle here was terminated about 14 to 21 days before planting, which is generally recommended. But you look at the differences in biomass. More biomass means more nitrogen uptake, means more soil coverage, means more carbon going in the soil. So if you wait until after corn planting to terminate your rye, you're looking at about double the amount of biomass. You're looking at about 30 kilograms per hectare more of nitrogen uptake. But when you wait to terminate your rye until after planting, a lot more issues can arise too. And that's kind of highlighted here with this carbon to nitrogen ratio. Significantly increases as well, which can result in some nitrogen issues early on in the season for corn. And some of the other issues too, when you just have that high amount of biomass with that rye cover crop, can be seedling disease. Here's a picture on the left here. This is some plants we dug in Lexington. You have that more biomass on the soil surface, the soils are cooler, you got more moisture, so you can have higher seedling disease instances. 2 nitrogen issues, which I mentioned earlier with the high seed-to-N ratio. So rye here on your left, you can kind of see it's a little more yellow, it's a little smaller. It just has higher stress early in the season with the more biomass you have. And also pests too. So again, more biomass, more pests can move in, whether it be slugs, whether it be animals. We saw that a lot throughout the last couple of years. So the objective of this trial was we wanted to evaluate the corn growth and grain yield following different rye cover crop termination timings. And also we wanted to try and understand whether or not we can kind of alleviate some of the issues in corn following these different termination timings by using both in-furrow fertilizer and also in-furrow fungicide treatments that we applied directly through the planter. You can kind of see the tanks here in the back of this planter. That's basically applying those products directly on the seed as we move across and plant in the field. So we had 3 locations across the state. Lexington was no-till. That was an irrigated site following soybean. Glendale, Kentucky was an on-farm location. That was no-till, rain-fed following soybean. And then Princeton out in far west Kentucky, no-till, rain-fed following corn. So we really try to capture a lot of different environments, a lot of different rotations too throughout the state for this study. So this study was a split plot. Our main plots was that rye cover crop termination. We used a rustic cereal rye, which is a very common rye cover crop variety. And we had 2 different termination timings. So chemically terminated about 14 to 21 days prior to corn planting. That's generally what is recommended to farmers. And then chemically terminated 1 day after planting. So that is kind of a newer movement with farmers where they're planting green and terminating that rye cover crop about at the same day or 1 day after planting. And then in-furrow starter was the second factor in the study. We had 3 in-furrow starter treatments. So 1030 furrow or ammonium polyphosphate, nitrogen and phosphorus fertilizer, headline fungicide, which is strobilurine fungicide, and then a combination of both of them tank mixed and applied together. So this figure here really illustrates what we wanted. We wanted to track differences in soil moisture throughout the entire corn growing season. So we buried soil moisture sensors about 4 inches deep into the corn rooting zone. So we have volumetric water content here on the y-axis. We've got the entire corn growing season, the date on the x-axis. And what we use to track this, we actually use generalized additive model analysis. So we can actually model the moisture response throughout the season and then use those models to tell us where is significant differences occurring in the different termination timing treatments. So different colors here, we got 1 day after planting, no cover crop, and 21 days before planting. So these bars up here, these horizontal bars, tell us where significant differences occurred. So if you look at, say, this bar here, where we have the blue is greater soil moisture. So, and that is equivalent to the growing season date down here. So 1 day after planting during this time of the growing season had greater soil moisture than no cover crop. This red part here means statistically less soil moisture, and that's equivalent to this area in the corn growing season. And really what we wanted to point out here is that early in the season with cover crop, especially 1 day after termination or 1 day after corn planting, we have statistically higher soil moisture. As that corn breaks or that rye breaks down throughout the season, it kind of goes away. We generally don't see as much difference in soil moisture later on in the season. So the later a rye cover crop was terminated, the greater the biomass which was produced. Therefore, we had greater soil coverage, less soil moisture evaporation. So the greater the early season soil moisture levels were observed, specifically in the corn rooting zone. That was as deep as we went, that 10 centimeter depth. Now, in addition to soil moisture, we also wanted to look at soil nitrogen. So we had total soil organic nitrogen. So that's ammonium and nitrate on the y-axis here. X-axis, we have the different locations, Glendale, Lexington, Princeton, and then we separated by the different termination timings. And kind of what was really interesting to us and kind of surprising to us was looking specifically at the 1 day after planting termination timing of that rye cover crop. Specifically at Glendale, so you look at the difference between that 21 days before planting and the 1 day after planting here. It's not statistically significant at Lexington and Princeton, but you kind of have the same trend where that soil inorganic nitrogen, these were sampled right before corn planting. So that inorganic nitrogen was actually higher in that late terminated rye. That late terminated rye usually took up a lot more nitrogen, but it was interesting to us that soil nitrogen was actually higher. We actually think maybe that immobilization of the inorganic N is occurring much later after termination. So about 21 days after termination, that immobilization maybe is actually occurring at the time of corn planting for that early terminated rye. And that rye that's still actively growing, not really incorporated in the soil yet, hasn't really transitioned to immobilization yet, why we're seeing some higher soil nitrogen levels in those treatments. So next here, this figure illustrates corn plant stand. These are the stand counts we took after corn planting. So we look at corn plant stand on the y-axis here. Again, we have those different rye cover crop termination timings, and then the different locations, Glendale, Lexington, and Princeton. And this is the interaction of plant stand and the termination timings and the in-furrow start. So in-furrow starter is the different colored box plots. So red is no in-furrow starter, green is fertilizer, blue is fungicide, and purple is the combination of the two. Really what we wanted to point out with this plant stand data,
10 minutes.
specifically the 1 day after planting termination timing. So specifically at Glendale and Lexington, just dramatic yield or stand reductions from the late terminated rye. And that really stems back from just the high amount of biomass we had and the difficulty of the corn plant getting through that high amount of biomass. So we see pretty dramatic reductions in that corn plant stand. Princeton, the biomass wasn't as big in Princeton, so we didn't have this significant reductions in the plant stand at that location. Now next, very similar to the last figure, but this is corn grain yield. So we have corn grain yield on the y-axis here. On the x-axis, we have the different termination timings. And then again, the box plots illustrate the in-furrow starter treatments. And really similar to what we saw with the corn plant stand, especially at Glendale and Lexington, we see those high reductions in plant stand equals high reductions in corn grain yield. So that really contributed to both Glendale and Lexington, and why we saw significant reductions in grain yield with that late terminated rye cover crop. Princeton, both cover crop treatments reduce yield. Princeton, again, was corn after corn. We think the nitrogen stress, because it was corn after corn, the high amounts of carbon in that soil with the rye, regardless of termination timing treatment, really drove the yield reductions that we saw in, and compared to the stand loss reductions we saw at the other two locations. And also too, what we really want to point out is the in-furrow starter, both on plant stand and yield. We aren't seeing really any effects in any site year from those in-furrow starters at all. So in conclusion, a late terminated rye cover crop can increase biomass contributions. It takes up more nitrogen. It helps conserve early season soil moisture. And we actually saw that it helped improve inorganic N levels right before corn planting. But the late terminated rye, because it gets so big, you can see in this picture just how big it was. Rye cover crop can actually reduce corn plant stand and yield. In addition, we also determined that the in-furrow application of fertilizer and fungicide really can't alleviate any of the negative effects of a late terminated rye cover crop. We think, one, the in-furrow, we're kind of limited on how much nitrogen we can apply. So I think moving to a starter where we can apply more nitrogen is important. Our corn seed was treated. We didn't see much seedling disease at all across the 2 years, so seed treatment may have been sufficient in these last 2 years. Then also looking at future research. I think looking at corn planter settings and equipment such as row cleaners, seedling depth, down pressure, stuff like that is a lot more important for managing corn following rye instead of trying to incorporate different in-furrow starter treatments. So with that, I'd like to thank the Kentucky corn growers for funding this trial for the last couple of years, and then my advisors, Chad and Hannah, and then as well as everybody. So many people help with a lot of these projects, so thank them as well. I think that's it.
Good. Thank you, Tim. All right, we have time for probably one question. It looks like we have one in the chat. And the 21 DP DBP termination, do you think nitrate has leached below 30 centimeters rather than being immobilized? Did you measure soil nitrate levels throughout the growing season as well?
That was one thing I wish we would have, but we didn't. We didn't measure it throughout the season, so you can't definitively say that the differences with the two timings throughout the season. It very well could have leached beyond the 30 centimeters, but it's kind of speculatory on why we saw that. It was kind of surprising to us that the nitrate was higher in that late terminated rye because it uptook so much more. But yeah, it definitely could have leached beyond there. That's another reason as well.
Okay. Thank you, Dan. I think that's all the time we have for questions. We'll be moving on to our final speaker of the session, Andrea. Are you here, Andrea? Oh, there you go.
Yes, can you hear me?
Yes.
Okay.
Okay. So the speaker is Andrea Keeney, and she works for Dr. Bailey, and she'll be presenting on the 2020 Angular Leaf Spot Observational and Field Studies. Okay, whenever you're ready.
Right. Thank you for the introduction, Jared. And like you said, my name is Andrea Keeney. And today I'm going to be talking to you all about some 2020 studies that we had going on with Angular Leaf Spot. We had a new observational study that we started in 2020. And then I'm going to highlight one of our field studies that we conducted at Murray State University. Okay, so just to kind of give you all a background about Angular Leaf Spot, or ALS, it is known as Pseudomonas syringae pathovar tabaci, and it is the most significant foliar disease in dark tobacco productions to date, and it has been since 2015. And we have had field trials going on, field spray trials with many different products since 2015, and with a lot of help from Emily Pfeiffer. But there's one main control mechanism for this bacterial disease, and that is streptomycin. But there has been documented resistance to streptomycin. And Brenda Kennedy at the Point Disease Diagnostic Lab in Princeton, she has received 101 samples in the lab, and she has screened those, and of those, 24 have shown resistance to streptomycin. Another study I just want to kind of mention is we did a 2020 Angular Leaf Spot variety trial in coordination with Zach Hansen at the University of Tennessee. Because there are no tobacco varieties that have any resistance to Angular Leaf Spot. So through this study, we're hoping that we can see that some varieties are less susceptible to Angular Leaf Spot rather than other varieties. I do want to mention going into this Murray field study that we do inoculate our plots approximately 5 weeks to 6 weeks after transplanting. We just want to make sure that those plants are about basketball size when we go in to inoculate. We do put out one spray application of the treatment prior to the inoculation. And then according to whatever our protocol is, we will then follow that for once a week after inoculation. So first I want to talk to you all about the yield for our Murray study. So across the bottom here, we have different treatments. We have our untreated control. We have streptomycin, which is the grower standard. And then we have 3 different copper products that we included in this study, and all are labeled for tobacco production. We have Nordox, COP5, and Sueva. You can see next to those treatments, we have either a 5 or a 3 in parentheses, and that is going to indicate whether that treatment received 5 spray applications versus 3 spray applications. We did that to see if we would be able to see any differences between a 5 spray versus 3 spray treatment, and we did here. So across the entire trial, we saw some significant differences, but we did see numerical differences in yields between the 5 spray and the 3 spray treatments. But what I really want to focus in on here is our Nordox treatments. So Nordox is a product that we've seen a lot of promise with in the past. So here you can see that numerically the Nordox treatments, both the 5 and 3 sprays, did have numerically higher yields than the other treatments. But going into our quality grade index, just to kind of give you an idea of what quality grade index is we have a federal grader come in and they assign cured leaves with a grade, which is normally, or which is alphabetical. We then convert that to a numeric number, which is easier for us to see differences in quality grade index. And also a higher quality grade index has the potential to influence price with a higher price when these growers take their product to sell it. But again, we do have our treatments along the bottom here. And overall, the 5 applications had numerically higher grade indexes than the 3 spray applications. And then we did see a significant difference between the 5 and 3 spray applications in our SWAVA treatment. Again, I want to focus in here on the Nordox. We see significantly higher quality grade index with our Nordox treatments than we do with some of our other spray treatments. But I do want to state here, I didn't show any ratings for this study just due to time and going through all of those. But we did see numerically lower ALS symptomology in all of our treatments compared to our under control here. But the main thing that I want to focus in on today is this new observational study that we started in 2020. So this kind of stemmed from a project that Emily did in her PhD, and she did it on onions, but we've incorporated it into a tobacco production system. So Andy kind of took the lead on this. This is a snapshot of a study survey that we did. So Andy was in contact with county agents as well as just dark tobacco producers in the past that he had known that had had some problems with angular leaf spot. So we asked them a series of questions, and with that series of questions, we ended up having 30 different fields that we included in this study. These 30 different fields spanned across 7 different counties in Western Kentucky and Northwestern and Tennessee. And with those 30 fields, we had 19 different growers that we worked with. With these 19 growers, we set a maximum of 2 fields per grower. And if that grower did have 2 fields, we made sure that they were a mile apart from one another for to be included into this study. But with all 30 of these fields, we were interested in a variety of variables. So I'm not going to go into all of these, but I want to kind of highlight some of them. So we're looking at rainfall amounts at these different fields. We're also looking at tissue in the soil as well as in the we're looking at nutrients in the soil as well as in the tissue. Again, looking at variety. We're just looking at the just general topography of all of these different plots, and I'll get into plots here in a minute, but just general locations of these fields. Looking at fertilizer application, more specifically, we want to look at nitrogen application, whether it's applied all up front or a split application of nitrogen. Also, we looked at pesticide application, and we looked at greenhouse management. Greenhouse management, we were really interested in the temperature that these growers are keeping their greenhouse at, if it's a constant temperature the entire time the plants are in their greenhouse, or if they decrease the temperature after these plants germinate. But I do want to say here, we didn't ask these growers to implement anything into their study that they normally would. So they were told to manage their fields as they normally would, and we would just come in and observe what they were doing. But just to kind of give you all an idea of how we laid these studies out in these fields, we did have 3 different plots per field. They were A, B, and C plots, and we tried to make them representative of the entire field. So some of these fields ranged all the way from 5 acres to 70 acres. So when we got into these 70-acre fields, it was a little bit more challenging, but we did a good job of just making sure these plots represented the entire field, whether that be next to a creek, a tree line, in a low-lying area, or on a hill. And then in plot B, I want to highlight is where we place our sensors. So we placed our soil and air temperature sensors, and then our rain gauges. We placed them right in the plant row so they wouldn't be in the way of the grower or their workers. And we buried our soil sensor, our soil temperature sensor, about 3 inches below the soil surface. And with those plots, we came back 3 different times throughout the growing season, and we did different things at each visit. So just to kind of highlight some of the things that we did. At our first visit, we laid out our plots and took GPS coordinates. We took these GPS coordinates not only to help us find these plots when we came back and the tobacco was the size that was in this picture, but we also want to look at different soil types that are in each plot in each field. We pulled transplant and soil samples and then placed our sensors. But to talk about the transplant samples for a second, so we've had some growers ask us if there's the possibility of this bacterial disease coming out of the greenhouse. So currently we're developing some PCR primers to carry out this analysis with to see if the the bacterial disease is potentially coming out of the greenhouse. We don't think that is possible. That is because we normally don't see a AOS symptomology in the field till about 5 to 6 weeks after transplanting. But at our second visit, we took a tissue sample, I rated for disease, and then I looked at weed pressure, insect pressure, and then if we saw AOS symptomology, we placed a rain gauge, as you can see in this picture here, right in the plant row at topping height. And we did not place rain gauges at each individual field. We placed rain gauges in an equal number of fields that had AOS symptomology and did not have AOS symptomology. So not all fields received a rain gauge. But at our third visit, we took green weights, as you can see in this picture here. We just cut 5 plants per plot and we just weighed those. But I am in contact now with growers to try to get a final yield for their fields here that we can possibly incorporate into our analysis. And then I took some final ratings. And then if I did see AOS symptomology, I took a leaf from that plot and I took it back to the lab in Princeton. And with a lot of help from Brenda Kennedy there, we collected lesions from individual leaves. We then confirmed that those lesions were Pseudomonas syringae pathovar tabacci. And then we screened those isolates for resistance to streptomycin and Nordox. So just to kind of lay out this picture here, if you're unfamiliar how these plating studies work, these plates were streaked with Pseudomonas syringae pathovar tabacci and was allowed to dry. And then you'll see here that there's an O and S
10 minutes
on each of these plates. The top is one isolate, the bottom is a different isolate. We did do 3 replications of each. So we soaked paper filter discs in the solutions of streptomycin at 200 parts per million and Nordox at 5 pounds per acre, which are grower standards for both of those. And then we placed those on these plates and allowed 24 hours to pass. We came back in 24 hours and is how we determined if these products were sensitive or non-sensitive is the clearing and inhibition around the disc. So you can see that all of these plates have clearing and inhibition around the disc for the streptomycin treatments. We didn't see that in the Nordox treatments, which was really surprising to us because we had seen promising results of Nordox in plating studies in the past. But that is something that was brought to our attention and we're working with now. But we do know that certain products work differently in a natural environment versus in a controlled environment. But just to kind of summarize like the observational study, so only 4 of my 30 fields had AOS, which was surprising to us because of all of the rainfall we had this summer. But 1 of my 4 fields did have streptomycin resistance. So I collected 3 different leaves from that field in each individual plot, and all 3 of those isolates came back resistant to streptomycin. But when I contacted the grower to let him know, he told me that they had not applied streptomycin to that field in over 10 years. So that is another question that was brought to our attention. But we do have some conflicting copper results. So like I said, we're going back and we're looking at what could be going on there. But there's a lot of information here and a lot of data. So we're currently analyzing that to see if we can find any kind of correlations here. But we're looking forward to a second year of this study. Not only myself and Andy and Emily, the growers are as well. And this project would not have been possible if it wasn't for the county agents that we worked with, as well as just the growers. If it wasn't for them letting us utilize their fields, this study wouldn't have been possible. And like I said, they're very excited about this study. But I also want to thank Altria for funding the project, and I will be glad to take any questions.
Thank you. Okay, we have time for questions. Looks like we have a question in the chat. The comment is, very interesting and important study. According to your work done, what are your perspectives on relative importance of multiple factors, climate, soil conditions on ALS? Any possible solutions and future plan?
So we've been talking to Dr. Wren to possibly work with a modeling study here to look at different weather patterns. So that's something that we're going to start here soon, is looking at all these different variables and then coordinating with Dr. Wren to see if we can possibly come up with a model for this specific project.
We have,
There's a question before that too.
there's, given the cost of making additional passes with equipment using additional product, would you recommend producers use 3 passes, even though 5 passes resulted in greater yields and better ratings?
I'm sorry, can you read that again? I can't see the questions.
Given the cost of making additional passes with equipment and using additional product, would you recommend producers use 3 passes, even though 5 passes resulted in greater yields and better ratings?
So yeah, we do know that with damage to the plant, that gives the bacterial disease a way in to the plant if there's a wound that occurs when they're going through the fields. But these growers are making way more than 3 to 5 treatments with just streptomycin in general. They have their own programs that they follow. In many of their programs, they're putting out way more than just 5 or 3 spray applications of streptomycin throughout the year. And then they have growers going in and they're in there, they're topping, they're suckering, and they're cutting. So there's a lot going on in the field besides just spray applications. But like I said, some of these growers are going in way more than just 5 or 3 times throughout the growing season. And we included that 3 spray application in also to see if we would see differences in copper residues. And currently we're working with Altria to get those results to see if we could see differences in the residues between the 5 and the 3 spray applications.
Okay, well, there is another question, but I don't know if we have time for that. Because 15 minutes is up, but so I can just hand this over to Jason.
Thank you.
Yeah, we're going on break.